Hey, everyone. Let's be honest. Genetics can feel like a total maze, right? Especially when you're trying to figure out how it all connects to what you'll actually see in the clinic. So, the goal today is simple. We're going to build a solid mental framework, like a blueprint, for classifying genetic diseases. Think of it as a map you can use from day one all the way to your final rotations. Ready? Let's jump in.
So here's our game plan. We're going to start with the big picture, get our main categories down. Then we'll dive into the large-scale chromosomal disorders. After that, we'll zoom in on single-gene mutations. First, the ones that play by the rules, and then the really interesting ones that don't. And we'll wrap up with that complex world where our genes and our environment intersect.
All right, let's start with that big picture. I want you to think of this as a master filing cabinet for every genetic disorder out there. Every single condition we talk about today is going to fit neatly into one of these drawers. If you can get this organization down, you're already halfway there.
So what are we even talking about here? At its core, a genetic disorder is just any health problem caused by a glitch in our genome. Now, that glitch could be tiny, like a single typo in one gene, or it could be massive, like an entire chapter of our genetic instruction book getting ripped out. This one definition really covers the whole spectrum.
Okay, let's start with the biggest possible errors, the problems that happen at the level of the entire chromosome. This isn't just a single typo. This is like a whole chapter of the instruction manual being missing or duplicated.
The first way we split these up is pretty straightforward. Does the error involve one of the main 22 pairs of chromosomes, the autosomes, or does it involve the X and Y sex chromosomes? So on the autosomal side, you have things like an extra copy of chromosome 21 causing Down syndrome, or a piece of chromosome 5 getting deleted which causes Cri-du-Chat syndrome. And when the error is on the sex chromosomes, you get conditions like Turner syndrome or Fragile X.
Now let's zoom in. We're going from the huge chromosome level down to the level of a single gene. These are what we call Mendelian disorders, named after Gregor Mendel, of course, because they follow these really predictable inheritance patterns that you can often trace right through a family tree.
So how do these single-gene problems actually work? Well, the first big split you need to know is dominant versus recessive. For a dominant disorder, you only need one bad copy of the gene, just one, and that's enough to cause the disease. But for a recessive one, you've got to get a faulty copy from both parents. And that is so important clinically. Why? Because it means the parents are often totally healthy carriers. They might have no idea they're even carrying a risk to pass on.
This right here shows you the odds for an autosomal dominant condition perfectly. If one parent has it, every single child they have has a 50-50 shot of inheriting it. It's a coin toss, really, but one with huge consequences.
A classic example you have to know is Marfan syndrome. This is a defect in the FBN1 gene, which messes with the body's connective tissue. Clinically, you're looking for patients who are very tall and slender with unusually long fingers, a sign we call arachnodactyly. But the real high-yield risk here, the thing you can't miss, is what it does to the aorta. It can get weak and dilate and even rupture, which is a massive, life-threatening emergency.
Huntington's disease is another key dominant disorder. It's a truly devastating neurologic condition, and what makes it so challenging both clinically and ethically is its late onset. The symptoms often don't show up until someone's over 40. That means they might have already had kids and passed the gene on before they even know they have it themselves.
Now let's flip over to recessive. See the difference? If both parents are carriers, there's a 1 in 4, or 25% chance, that their child will get both bad copies and actually have the disease. There's also a 50% chance of having a child who's a carrier, just like them. Okay, 2 high-yield examples for you here. First, cystic fibrosis. That's a defect in a chloride channel, which leads to that trademark thick mucus and secretions. And then there's phenylketonuria, or PKU. It's an inborn error of metabolism. And this is a fantastic example of modern medicine in action because we test every single newborn for PKU. Why? Because if we catch it early and put them on a special diet, we can completely prevent severe brain damage. Lastly, for Mendelian patterns, we've got X-linked recessive, which has this really unique crisscross pattern. Just think it through. A father gives his Y chromosome to his sons and his X chromosome to his daughters, right? So if a father has an X-linked disorder, he can't give it to his sons, but 100% of his daughters will get his affected X, which makes them carriers. Then, those carrier mothers have a 50-50 chance of passing that bad X to their own sons, who will then get the disease. This is the pattern you'll see for diseases like hemophilia. Okay, so we've set up the rules: dominant, recessive, X-linked. But, in biology there are always exceptions to the rules. So let's look at some really fascinating single-gene disorders that just don't play fair. First up, mitochondrial inheritance.
You gotta remember that our mitochondria, the powerhouses of our cells, have their own separate little loop of DNA, and we get it only from our mothers. So a mother with a mitochondrial disorder passes it to all of her children. And since this is all about energy, you usually see problems in tissues that need a ton of it, like nerves and muscles. The severity can also be wildly different between family members because of something called heteroplasmy, which is just a fancy way of saying there's a mix of good and bad mitochondria in the cells.
And here's where things get really cool, because science is never, ever set in stone. For decades, the central rule was that mitochondrial DNA comes only from your mom. That's it, full stop. But look at this. Brand new research is starting to show that paternal DNA might actually influence it after all. It's just a great reminder that the facts you're learning right now, they're constantly evolving.
This has to be one of the most mind-bending concepts in all of genetics, genomic imprinting. What we have here are two totally different syndromes, Prader-Willi and Angelman, and they're caused by the exact same deletion on chromosome 15. So what's the difference? It's all about who you got it from. If that deletion is on the chromosome you inherited from your dad, you get Prader-Willi. If it's on the one from your mom, you get Angelman. It's because the other parent's gene is imprinted, or basically switched off, so the one you inherit is the only one that counts. Just wild.
Okay, we've done whole chromosomes and we've done single genes. But what about most of the common diseases you'll see every day? Well, for that, we need our final category, the one where lots of genes meet the outside world.
You know, the most common diseases, things like hypertension, diabetes, most cancers, they aren't caused by a single bad gene. They're multifactorial. It's this super complex dance between dozens, sometimes hundreds, of different genes that each add a tiny bit of risk, all mixed together with environmental factors, like your diet, your lifestyle, what you're exposed to. That's why they're so common, and it's also why they're so hard to predict.
And speaking of those environmental factors, some are so powerful during development that they can cause birth defects all by themselves. We call these teratogens. And this list right here is absolutely need to know for any medical student. Things like alcohol, drugs like thalidomide, certain infections we group as TORCH, and radiation. All of these can seriously disrupt fetal development.
But with teratogens, timing is everything. If an embryo is exposed really early, before the 3rd week, it's usually an all or none situation. Either the damage is too great and the pregnancy is lost, or it manages to recover completely. The really critical window, the most vulnerable period, is from the 3rd to the 9th week, especially weeks 4 and 5. That's when all the major organs are being formed. After that, in the fetal period, exposures are more likely to affect overall growth or just injure organs that are already there.
All right, let's wrap this up with a super quick recap. We built our framework starting with those 3 main buckets: chromosomal, single gene, and multifactorial. Inside single gene disorders, we have the predictable Mendelian patterns and those really cool exceptions that break the rules. And we ended by seeing how genes never work alone. The environment and what we're exposed to plays a huge part.
So I'll just leave you with this to think about. The next time you're in the clinic taking a family history, don't just ask what diseases people had. Start thinking about how they might be inherited. Are you seeing clues of a dominant pattern, maybe a recessive one? This framework isn't just for passing an exam, it's a real clinical tool that will help you understand your patients on a much deeper level.